What is a Punnett square? So in the last lecture, we went over all the basic information about who Mendel was, what his laws were, and many of the vocab words needed for this unit. In this video, I just want to quickly go over an example of how we can set up and use a Punnett square to determine the likelihood of certain offspring having certain traits. So all a Punnett square is a chart that helps us predict the probability that an offspring with a certain genotype and a certain phenotype will be produced. So it's the likelihood of two parents with brown hair having a brown-haired child versus a blonde-haired child. So to make a Punnett square, the first thing you do is you just draw a box, and you divide it into 4 parts. So you just make 4 even little boxes. So after you have your box drawn, you just label the left-hand side of the box with the genotype of one parent. In this case, we'll be looking at flowers, red versus white flowers. So let's say we have one parent that is heterozygous, so it has one dominant, one recessive. So the dominant, the capital R, stands for red. The lowercase r is the recessive allele, which is white. And it does not matter which parent goes on which side of the Punnett square, as long as you have one parent on one side and one parent on the other side. So you can see we put the big R and little r on the left-hand side of this Punnett square. We then take the genotype of the second parent, and we put them on the top, one in each column. So let me just highlight something. So we talked about in the last lecture about the law of segregation. Each one of these rows and columns, this, and this, equals one gamete, one sperm or egg cell. So you can see this is the alleles at the top represent the law of segregation. This is one allele, this is the other allele. So they segregated. This one, he had big R, little r, they segregated. So the separation of the alleles on the side of the Punnett square represent
now we just fill in the
law of
boxes.
segregation. So what we do is we fill in the columns and the rows with whatever letters in that row or column. So we fill in all the r's this way, we fill in all the little r's that way, big r's go this way, little r's go this way. And we see in this first box we have big R, then little r. The box underneath that we have little r. The box to the top right we have big R, little r again, and the final box at the bottom we have little r. So you just fill in the boxes, which whatever letters above it. Alright, so after you fill in your boxes, that's great, but we kind of want to determine what they actually mean. So each individual box in a Punnett square represents a baby, represents an offspring. So they represent the genotype of that offspring. So if you look at the previous Punnett square, you could see that we had 0 big R, so we had no homozygous dominant. We had 2 big R, little r, so we had 2 heterozygotes. And we had 2 little r's, so we had 2 homozygous recessive. So those are the genotypes. Remember, the genotype are the 2-letter combination of the 2 different alleles found in the offspring. After we figure out the genotype, we then interpret the genotype to figure out the phenotype, which remember is the physical characteristic of the organism. In this case, the capital R stands for red, the lowercase r stands for white. So the 2 heterozygotes will be red colored because they have a dominant allele, and the 2 homozygous recessives will be white. So the cross between these 2 parents produced 2 red offspring and 2 white offspring. Or they should if you have 4 offspring only. In other words, it's a 50-50 ratio. Alright, so let's do a practice problem together. So a cross between a female with straight hair, HH, and a male with curly hair, little h. So we want to figure out what the genotypes are, the phenotypes are, and the ratio of them. Alright, so let's deal with the female first. Doesn't matter which side of the Punnett square you put her on. We'll put her on the top. So we separate the alleles. First H goes there. Second H goes to the next column. Now we have the male, which is homozygous recessive. Little h goes there. Little h goes there. And now we just fill in the boxes straight down. Big H, straight down, big H. Now we follow the little h across to the right, little h there, little h there, little h there, and little h there. So genotypes, it wants the ratio of homozygous dominant to homozygous recessive to homozygous dominant to heterozygous to homozygous recessive. So we look at our 4 boxes. None of them have big H, so we know the ratio is 0, homozygous recessive, dominant. They are all big H, little h, so we have 4 heterozygotes, and then we have none of the homozygous recessive. So now we look at the phenotype, straight to curly. So the straight hair must be dominant because they're represented by the 2 big H's. So therefore, all 4 of our offspring have a dominant allele, are heterozygous. Therefore, we're going to have 4 straight-haired children, and we have no curly-haired children. So the purpose of this Punnett square is to see what the likelihood of a certain phenotype appearing, or a certain genotype appearing. In this case, you see if these 2 people were to have a child, 0% of the time would their child develop curly hair. All of their kids, 100% of the time, would have straight hair. Alright, so another example between a black dog that is heterozygous and a brown dog that is homozygous. So black must be dominant, brown must be recessive. So we fill them out again. Big B, little b, little b, little b. Fill them down, fill them down, fill them across, fill them across. So now we look at the genotypes in the boxes. Each one represents a child, an offspring. There are no homozygous dominant children. There are 2 homozygous or heterozygous children, and there are 2 homozygous recessive children. So now phenotypes, black versus brown. 2 of the children are heterozygous, so they're going to be black. The other 2 are homozygous recessive, so they are brown. So it's a 1/2, 1-to-1. We can reduce that to a 1-to-1 ratio. So 50% black, 50% brown. Remember, both these genotypes always look like the dominant phenotype. Alright, last one. Practice problem between a yellow corn and another yellow corn. So, and the dominant is yellow, the recessive is white. So we do it again. Separate the alleles due to the law of segregation, Mendel's law. Drop them down. Whoops, that is a Y, not an X. Drop them down. Drop them across. Drop them across. So genotypes, we see we have one of them that is homozygous dominant, this one right here, top left. We have 2 that are heterozygous, and we have 1 that is homozygous recessive in the bottom right. So we have a 1-to-2-to-1 ratio of homozygous dominant to heterozygous to homozygous recessive. For phenotypes, look at the colors. These first 2, this one and this one, both appear yellow. So we have 3 of them are yellow, and 1 of them is white. This is actually a very, very common example where you have 2 heterozygotes you're breeding together. The ratio will always be, in genotypes, 1-to-2-to-1. In phenotypes, the ratio will always be 3-to-1. And if you do enough of them, eventually just remember those ratios. And this is the last practice problem. So you're also in class given a sheet of practice problems that you're to work through. Refer to this video, refer to your textbook in Chapter 6. If you have any questions, feel free to email me. I'll do my best to help you out. If you can't remember anything, make sure you review this video or Google other videos.